Okay, great. So this session is really characterized by great geographic diversity. So we had one continent represented, Asia, in the first topic, and now we'll have Europe represented in the second talk. This will be from Zach Mainen, who is from the Champalimaud Center for the Unknown in Lisbon, Portugal. And Zach will tell us today about serotonin and the regulation of neural inference and learning.
Thanks for inviting me, and thanks to everyone for being here in the audience. So my brief version of my history is about 30 years ago, I was super interested in artificial intelligence, and there was a boom very much like this one. And then somehow I ended up in a neuroscience lab. I have no idea how. And the rest of my career, I've been worrying about what I thought at the time was a bunch of goo. And it's kind of interesting to look at it now from the perspective of this meeting and try to start with a little bit of that perspective. So my lab works on decision making, and we're particularly interested in serotonin. Serotonin, as you probably know, is involved in the treatment of depression. So this is Prozac, a serotonin reuptake inhibitor, and it's used to treat depression. There's approximately 300 million people with depression in the world. About 10% of you are, statistically speaking, going to be depressed. And suicide is the second leading cause of death amongst people of your age, rather surprisingly. Yet, although serotonin reuptake inhibitors has got a large market share, we don't really know how they work. Augmenting the mystery, the psychopharmacological mystery of serotonin, is somewhat well-known involvement of serotonin in consciousness-altering drugs like LSD and psilocybin, which we also don't know much about how they work. And even more puzzlingly, perhaps, these drugs are now undergoing clinical trials to treat depression, which seems strange if what they do is alter consciousness and what depression is something to do with mood. So there's a great deal of interest and confusion from the pharmacology side. And that's a lot why I've been interested in this. So, are neuromodulators an example of canonical computation, or are they just a quirk? Could we have depressed robots? Does that make any sense? Is there something like a serotonin that's going to be necessary in a machine? That seems a bit strange. But on the other hand, could intelligent machines tell us something about depression or consciousness? Also seems a bit odd. So when asked to prepare this talk, I actually was in the mood of saying, well, you know, brains and machines have specific hardware constraints, evolutionarily or in terms of engineering. Some of this is all random. And some of these constraints are not shared, and we might actually learn very little from, say, machines about serotonin and depression. They don't need to have anything to do with one another. But on the other side, brains and machines solve similar problems. Some of the features of those problems will require similar solutions, and the canonical computations that are involved might very well involve neuromodulators, in which case we really ought to pay attention to machines and how they learn. So obviously we need to be a bit more specific, so let's think about what is a neuromodulatory system. First, neuromodulatory system is something that's distributing a signal very widely in the brain. This is a serotonin system. There are 10 subnuclei, but this one, dorsal raphe nucleus, provides most of the serotonin, and it innervates most of the brain, at least most of the cortex and the parts that attract a lot of our interest. So there's a very small number of cells that are clustered, so they are thought to broadcast a low-dimensional signal, not necessarily scalar, but they don't have bandwidth to broadcast a lot of distributed code. Also, perhaps, well, it goes without saying that this is a special transmitter, and that probably is why pharmacology is so interesting in the case of serotonin. If you bathe the brain in glutamate, which all the neurons in the cortex use, all the excitatory neurons use to communicate, you're going to get a very weird effect. If you use serotonin, you might mimic, to some degree, the natural behavior of the system. And then fourth, what we've realized recently, well, relatively recently, is that they're not modulators that act only on a slow time scale like drugs. They actually have fast actions. Their action potentials transmitting signals down those long axons, and as I'll show you a little bit later, those signals can act quickly on the brain. And so we've been able to study this, and a lot of what I'll tell you about is due to optogenetics, which I think all of you know, but allows us to use genetic tricks, viruses, transgenic animals, to introduce molecules into serotonin neurons and then control them on a rapid time scale with light. So we could control them with pharmacology, but not quickly. So with optogenetics, we can ask questions on a behavioral time scale, and we think that fast time scale is also very important to how they work, and that would never have been accessed only with pharmacology. So given those characteristics, what sort of candidate computations might be neuromodulators be involved in? Well, we've heard a few times about reinforcement signals, like the famous dopamine reward signal illustrated here, and like a previous speaker, I think it was Matthew Botvinnik. I don't have time to explain this, but hopefully you all know it. If you've heard this a few times and you don't know it, probably it's a good thing to go look up in the textbook. So that's one possibility, and it was once hypothesized by Peter Dayan and Nathaniel Daw that serotonin might be a complement to dopamine, delivering a kind of anti-reinforcement signal. A second class of possibilities that I think is quite interesting is something more like global or semi-global normalization. David Heeger and I were talking a little bit about this after his talk. These circuits, LTSMs and other sorts of circuits, often require some type of normalization to go on that isn't necessarily strictly local. And to do that, you might need a signal that was broadcast like a serotonin signal, for example. And then finally, Kenji started to talk about this, talked about this in the context of serotonin, but he really was one of the pioneers. I think he's the first person to introduce this idea that neuromodulators could be setting meta-parameters or hyperparameters for learning, or whatever hyperparameters are required in one's model. And often these things are tuned by hand, and we don't worry so much about them, but in an autonomous agent like an animal, they need to be worked out autonomously in the animal, and things like learning rate can be very important to how brains function. So to get into how we have been studying these things, one of the first types of experiments we looked at was a probabilistic reward task. And we wanted to know whether serotonin would bias the animals as if it was good or bad. So this is very similar to what Kenji just showed, where a mouse makes a choice between left and right, and it gets water rewards. And we change the probability of reward so that one side is high and then low, and then high again, so the rewards are flipping back and forth, and the animal has to learn to follow that. They're probabilistic, so it's got to integrate over time. And then in this experiment, we stimulated serotonin neurons in uncorrelated blocks to see whether it would bias the animal as would reward. Now, if you did this with dopamine, it would bias the animal toward the side where the stimulation was delivered. What we found when we stimulated serotonin was that the animal tracked the rewards as expected. So you can see it tracking the red dot here, but the effect of stimulation was effectively nothing. So here's a regression model, and you can see the effect of water on choice. You can see here's the effect of serotonin on choice. So this and several other assays like this have shown us that we get nothing that looks like reinforcement nor anti-reinforcement from serotonin. So we don't think that this is the right way to think about it. And this suggests to some degree that maybe thinking of serotonin as mood is not the right way to think of it. It doesn't seem to be good or bad for the animal to receive serotonin stimulation. So what about these other ideas? Well, there is a thread in the literature on serotonin that goes back a little while that's from Trevor Robbins' lab and collaborators like Rasha and Cools, who have shown that serotonin is involved in reversal learning. or what they call cognitive flexibility. So we've done recently similar experiments in mice. Those experiments were primarily in monkeys and in humans. And the task I'll take you through looks like this. Basically, you train the animal using, in our case, odors to expect either reward given odor 1, odor A, or nothing given odor B. So the animal has strong expectations about these odors, and then at the reversal step, you swap them so that A is now nothing and B is now reward. So the animal experiences a positive surprise and a negative surprise, either a disappointment or a anti-disappointment. What we're really interested to see is, what do serotonin neurons do? Are they similar to dopamine neurons or anti-similar? What kind of signal do they broadcast? So we used another type of genetic trick to introduce a calcium-sensitive fluorophore into the dorsal raphe neurons, along with another protein, fluorescent protein, that would let us make sure there were no movement artifacts, which turns out to be a bit complicated for this system. Then you have a fiber optic, and you can essentially collect the bulk signal from serotonin neurons while the animal does this reversal task. You could do this over weeks. It's quite stable because you're only recording a bulk signal, not individual neurons. So what did we see? So if you look at serotonin responses to the reward, after training, there's very little response. If you ask later, I'll explain why this is going downhill, but it's not important. So in an expected reward situation, serotonin neurons don't care so much. Then when the animal experiences a positive surprise, this is water now preceded by the odor that used to be not so good. Now it's surprised that it gets water. You get a nice response. This is very similar to what dopamine neurons do. And actually, we tested dopamine neurons in exactly the same task, and they also like positive surprises, as is well known from the work of Wolfram Schultz and many others. So, okay, that's cool. Maybe that means serotonin is somehow detecting reward or delivering reinforcement. No, so if we look at non-reward responses, so this is when we deliver nothing. Now we trace that outcome. At the beginning, after training, again, the serotonin neurons are not interested in nothing. But after the reversal, when they had expected water for the odor that preceded nothing, and they get nothing, they are actually excited by receiving nothing. So excitatory response to positive and negative surprises, basically this is a surprise or uncertainty signal. How do we think about that? Well, we like to think in terms that Kenji also started to describe, which is in terms of some type of inference. And this is a very simplistic way of looking at it. You can imagine that higher order cortical areas have expectations, and lower sensory systems and primary sensory areas, for example, have evidence or data. And those have to be combined to make inferences or compute posteriors. If you have a high confidence in your expectations, if your model is something that you trust, then you're going to weight these higher than the data, or relatively higher. Then if you have low confidence in your expectations, if, say, the world just changed and now things are not as you expected, you're going to downweight your the influence of your model and pay more attention to the data. So if serotonin neurons report surprise or uncertainty as they seem to, one would expect not an increase in priors, but a decrease. What's, of course, tricky is exactly where those priors might be. But one thing that we looked at was, what about learning rates? So if your model is wrong, one thing you need to do is to update the model. So in the same task that I just showed you, we performed chemogenetic inhibition of 5-HT neurons, of serotonin neurons. And what we saw was, consistent with previous results by the Robbins group and so on, serotonin blockade, chemogenetic inhibition, slows reversal learning. So without serotonin, the model seems to be updated less quickly. Now, if we go back to the probabilistic reward task, this is also a kind of reversal learning, right? Every time the reward switches from the left to the right, the animal should perhaps update what it thinks about where the good side is. So do we see such an effect in this task? Well, in the original paper, this 2015 study, we didn't see anything. Fortunately, we have an ongoing interaction with Peter Diane's group, and after presenting these data, one of his postdocs, Q. Iagaya, reanalyzed the data, and what he found was something that we hadn't really noticed. Turns out if you look at these choice and reward data, as I showed you, the animals are tracking very nicely. But if you look at the ITIs, there are trials in which the animal basically takes a little break and then comes back after a while. So the ITI might be 10 or 20 seconds, like in these cases. Whereas in most of the trials, the animal does it fairly rapid fire. So what Q did was to analyze separately the long ITI and the short ITI trials. And what he found is that after short ITIs, the behavior of the mice could be described as a win, stay, lose shift. Essentially, they only really made their next choice based on the immediately preceding outcome. If they got a reward, they stayed. If they didn't get a reward, they switched to the other side. However, if you look at the long ITI trials, then what he saw was a RL-like weighting of prior trials with a several trial history, with a tau that goes backwards. So this indicated that the mice use two different decision mechanisms. One is kind of a cheap shortcut, just repeat or not repeat, depending on how it went. And the other requires the integration of information, but it's slow and perhaps unflexible, as Kenji was also describing. So fitting a model in which two different mechanisms are responsible for the animal's choices, what we found was that the estimated learning rate for the RL value-based decisions is increased by serotonin stimulation from here to here, whereas the win, stay, lose, shift decisions are not influenced at all by serotonin. So it's fairly hard to see the effect unless you look at these long ITI trials, and then the effect is actually quite large. So this is consistent with the reversal learning data that I showed you, that again, serotonin seems to increase the learning rate, a kind of hyperparameter, metaparameter. So that makes sense. Serotonin neurons report surprise, and that sort of surprise tells them to update the model. But we know that serotonin has short-term effects, which I mentioned. Kenji described these effects on weighting. I actually don't have time to go into several of the behavioral studies that we've done to try to look at the effects on complicated behavior. I just wanted to show you a very simple behavior where you can see very dramatically how fast serotonin acts. So this is an animal running around an open field. The trace is the animal position, and the colored bits are when we stimulate. And this is the speed of the animal. So here's his baseline speed, and then we turn on serotonin, and within a second, the animal has, on average, dropped about 50%. It's quite dramatic effect, and it's quite large. So this is not a learning effect. This is not the animal learning over 1 second to slow down. There are clearly direct effects, as there are for dopamine, as well as learning effects. So I'm going to skip that. So what else could that be due to? Well, could we think of this kind of direct effect as an immediate change, for example, an increase or a decrease in the influence of a prior? In the case of an open field experiment, who the hell knows what the prior is? Who knows what the animal is doing? So I could perhaps account for things that way, but it's obviously very ambiguous. With these more constrained tasks, like the probabilistic choice task, you can start to get a better handle on priors, but it's very difficult behaviorally to really know what is a prior. You can make stories in which things go one way or the other. At least I find it difficult to constrain things that way. But there's another idea, obviously, go look in the brain. And so what about recordings? And we were inspired by this work of Mate Lengel, Joseph Fisher, and colleagues, who basically said, hey, look, spontaneous activity in the absence of sensory input, it would be equivalent to the prior. It's the expectation, the absence of evidence. So what if you look at the effect of serotonin on spontaneous activity and compare that to the effect of evoked activity? What's the effect? So we went to the olfactory cortex in the mouse. Now this is an anesthetized mouse, unfortunately, but very nice serotonin, well-controlled serotonin, and nicely recorded olfactory cortical neurons. We can ask, what does serotonin do to spontaneous versus evoked activity? And the result is that serotonin inhibits spontaneous activity quite dramatically. So here's a piriform cortex neuron. Here we start stimulating the serotonin neurons, and here's the neuron shutting off. It looks a little bit like the behavioral traces I showed you. If we now look at odor-evoked activity, we see that serotonin spares odor-evoked activity completely. So this green trace is spontaneous effect that I just showed you, and then if we have an odor, which is this response, plus the serotonin, all we really see is that the baseline is decreased, but if we subtract this out, so we compare just the odor to odor plus light, subtracting out the effect on spontaneous, there's really no effect, neither on the single neuron, neither on the population level nor on the single neuron level on the odor-evoked component of the activity. So, in our minds, this result, at least in piriform cortex, is again consistent with this idea that if serotonin neurons report surprise, then this could implement both a change in learning rate and a decrease in the weighting of the prior. Now, I would say there's still a fair bit to do to make this story airtight. I'm showing you pieces of data from different experiments, and it's possible that olfactory cortex is a different story than other bits of cortex. I don't know that these signals are global. There are a lot of loose ends, but to some degree, things hang together. I've indicated the learning rate, so that's a hyperparameter. That's one of the sort of traditional meta adjustments that you might make in learning. So that is not always been associated with serotonin, but that seems to be one of its roles. And as for normalization signals, well, that's in a way another way of thinking of downweighting the prior. It's a kind of way of normalizing part of the brain versus another part of the brain, or a way of globally scaling the activity in a particular region, because we, at least as far as we know, in a particular cortical region, serotonin would uniformly affect the whole population of neurons. Of course, that remains to be checked in detail. So how does that explain our conundrums of interest? What do we have to say about depression? So we did not propose this model. It's been out for a little while now. There is a line of thinking in the field that perhaps SSRIs do not change mood, but rather act via an effect on plasticity. So essentially, you start taking the SSRI, and it pushes you from a kind of low plasticity state in which you could be stuck in a depressed model, let's say, toward a more plastic state in which you could go back and forth. So to put it maybe in more relatable terms, imagine if something really bad happens and you've lost a loved one. You're now stuck in a bad model because you expect that person to be there and they're not. What you need is not necessarily a boost in mood. You need to adapt to the new reality. You need more plasticity, basically. You need to adapt your model. So perhaps SSRIs are acting in conjunction, usually with therapy, to help patients to basically adapt to the world rather than just pushing them toward being happy. So that makes a lot of sense with respect to the story that I've told you. I think there's a lot still to be done to change the thinking in the field toward this, but I think it's a very interesting way to think. And then as to these things, hallucinogens or psychedelics, what we see, according to the data that we have so far, is also consistent with the idea that these drugs might be acting by decreasing, or perhaps increasing, but decreasing priors. So it's kind of an interesting way of thinking about hallucination. If you think about hallucination as like dreaming, you might think a hallucination is when your expectations are overwhelming the actual data. And of course, in dreaming, there is not much data. So whatever models you have are generating activity, and that could result in imagery and so on. But another way to think of hallucination is normally sensory information is ambiguous. For example, many visual scenes are ambiguous unless you have a strong prior to constrain the data. If you cut your priors, if you drop your expectations, you may start to now see things in ways that you are not used to. You may not see them the normal way, but see them in another way which is equally consistent with the data. So this goes along with this idea of plasticity, dropping expectations. It all hangs together potentially. But again, lots to do to verify. We haven't actually shown that this is what happens with 5-HT2A agonists, but we're in a position to test that and to try to understand better how these fascinating molecules are working. And that is all I wanted to tell you. I want to end by thanking the people who did this work. So Iran is an amazing postdoc who now has his own group in Israel. Sarah is now a postdoc, was a PhD student, is now a postdoc at Harvard. And Patricia, another graduate student, is, we're trying to get back in Portugal. There are a variety of other people who contributed to the work, and our collaborators, Peter, Diane, and Kiyo Igawa, I think, as well. Thank you all for your attention.
Okay, thank you very much, Zach. And we have time for some questions. Be ready, Zach, because her questions are very good.
Hi. So there was a talk at CCN a while back about how acetylcholine corresponds to absolute value of prediction errors. And on the other hand, so Angela, you and Peter Dayan have this idea of a model of expected versus unexpected uncertainty, which requires a time horizon during which I'm monitoring the variations in reward. So on that note, two questions. First, what do you think is the role of expected variability in reward as opposed to either positive or negative prediction errors in serotonin? And second, the time horizon. How does depression either change that sensitivity to the variation or to the time horizon across which...
Depression?
Yeah. So, or using words maybe too equivalently, like suppressing or activating serotonergic neurons. How does that influence either the time horizon of estimating uncertainty, expected uncertainty, or basically how much variance there's tolerance for?
These are very good questions. You're right.
I told you.
Well, the short answer is we don't know, but let me speculate. So one thing that I think is relevant is that there are, you started talking about different modulators. So it doesn't look like serotonin has a monopoly on reporting uncertainty. So one possibility is that different neuromodulators specialize in different learning systems. Kenji talked about basal ganglia for one type of learning, cortex for a different type of learning. So there are differences in distributions of axons. There are relationships in the signals. So at least in my thinking, there's not going to be a neat one-to-one mapping between neuromodulator and parameter, more a complicated interrelationship. And then in terms of slow and fast timescales, it looks like these signals that we see are most prominent in these kind of abrupt changes where we'd call that unexpected uncertainty, right? So in this reversal learning experiment, the animal has been seeing the same odor paired with the same reward for days. And then we only once change it, and we get a nice big signal. So that's unexpected uncertainty. in the case of expected uncertainty, we haven't really done the experiment, but I believe the signals are smaller. There's some, I'm sort of reading between the lines with some experiments out there, but I would say it falls in that way. And then there was sort of a third part about that's enough. Okay, great. We can talk afterwards.
Other questions in the back?
Yeah, so this might be a little off the wall, but apparently there's some literature on lobsters, which also have a serotonin system.
Yeah.
But in the lobster, it's the one, they have like a little social hierarchy, and the ones at the bottom actually have very low serotonin levels, whereas the ones at the top have very high, and that seems almost to go maybe counter to the model you've presented, where you would think if you're at the bottom, you'd really want to update your model so you could try to climb up to the top, whereas if you're at the top, maybe your model's good and you don't need to update it. So I don't know if you can possibly explain that.
Well, the nice thing about serotonin is that you can frequently come up with either the explanation or its opposite, depending on... Well, it's a bit, yeah, I mean, a little bit sarcastic, but... you know, yes, there's also data even on these social hierarchy questions, even in primates, there's data on serotonin and social status. And in that case, generally, things go as higher serotonin. I think it goes the same direction.
Same direction.
Yeah, so higher serotonin, higher status. I don't have a ready explanation for that. It could mean that the theory is shot. I think the way I think about it is we need to look. Instead of looking at a whole bunch of diverse behaviors and physiological functions and seeing this kind of strange pattern, if we look at a slightly lower level of computation or algorithm, as I was trying to pitch. It's not quite the level of, say, just a transmitter to get from point A to point B. It's, say, a metaparameter or a normalization issue. It's not that clear how to map that onto each and every behavior. It takes a lot of effort to understand how, say, a normalization constant is going to play out in a particular task or social situation. So maybe that is, being a bit wishy-washy, but that's how I see it. So I think we're going to, eventually, we won't be able to sort this out at the behavioral level. We're going to need recordings, careful recordings with models and behaviors. So there remain serotonergic mysteries.
Terry.
So your interpretation of the serotonin stimulation experiment after it was reanalyzed is that it signals surprise and uncertainty and then changed the learning rate. But there's another interpretation, which is actually something that Kenji Doya actually suggested a long time ago, which was that it's the temporal discounting. So what happens when at the beginning it's basically greedy, right? That's temporal discounting of 0. You just basically are taking whatever is available that you have, and then if you're integrating over a much longer time, right, that's temporal discounting of close to 1.
Yeah.
Where you have to take into account a long history.
Yeah. And
so maybe it is surprise, and then changing the temporal discount factor.
Possibly. I now forget exactly all of the variations on the model that were run in that experiment, but there is ambiguity in these parameters, and we may not even have our models are wrong, then the parameters are wrong. So one of the other things that goes along with this is you study serotonin, you can sort of work on any model space that you want, and you're going to get slightly different answers. So yes, I think Kenji's insight that I'm certainly following in, and Peter's insight is if we think in terms of reinforcement learning, we get new hypotheses about how to think about these things, and even if the original assignment to variables is wrong, that's okay because we can try other ones. We don't want to only have correct hypotheses. We want testable hypotheses and new ideas, and that's what we got. So I think it's great. I think theorists shouldn't be too stuck to particular models, otherwise they'll just be spending half their careers being wrong, and you just have to move on. And experimentalists have less of a problem with that. They're used to being wrong all the time.
Very, very beautiful talk. I was I reminded myself while looking at your talk that there was this recent paper from Carlos Brody lab where inactivating the posterior parietal cortex makes the animals better in the task because they forget their priors. So I was wondering if you could think that maybe it's the serotonin projections into the posterior parietal that might be actually deleting these priors. So maybe using serotonin could also improve in a similar manner the performance on these animals, on these tasks where the past has no influence on the current.
It's a good suggestion. I agree. One of the tricks and tricky things when we start to look at those interactions is they're mostly bilateral, reciprocal, right? So it can be, for example, orbitofrontal cortex also has some interesting relationships to serotonin, and there's a two-way interaction. So could be that posterior parietal priors are something that drives serotonin and then gets affected by it.
Okay, great. Well, great talk, Zach. Let's thank him again.